Our specific product is fibrinogen-depleted. It's completely xeno-free, doesn't require any use of heparin. The FDA doesn't want people utilizing animal to human components. What's going to be the most efficient and effective product that's going to grow their cell types?
Thank you to the International Society for Cell and Gene Therapy for supporting Onyx Live. I am Federico Cittrich, reporting for Onyx Live here in Dublin at the ISCT. Today we are joined by Kim Brown, CEO at Compass Biomedical. Kim, good afternoon. Thank you for joining us.
Thank you. Compass Biomedical was started back in 2006. We started off as a company that utilized platelets for wound care, burns, sternal closures. After a while, around 2013, we decided to switch and utilize human platelet lysate for use with T cells, NK killer cells for use in the cancer therapies, as well as MSCs for the orthopedic field.
Kim, a lot of the attention goes to the final cell therapy product, but perhaps a bit less to also the materials used to grow those cells. So, based also on what Compass does, how much can culture components actually shape the quality and consistency of the final product?
That's a good question. It depends on what the customer is using the product for, what type of cells are being grown. So our specific product is fibrinogen-depleted. It's completely xeno-free, doesn't require any use of heparin. Other media similar to ours does use heparin, which could cause issues with precipitate in the final product with growing the cells.
You mentioned it, you also used xeno-free material, so why are developers moving towards defined human-derived and xeno-free materials in this context?
Sure. When people are growing different cell types, the cells end up getting washed and then injected potentially into patients for patient care, specifically being used in cancer therapies. The FDA doesn't want people utilizing animal to human components, so trying to step away from the animal sector and only have human to human when feeding those cells is more appropriate. So that's why we think with our platelet lysate, completely getting rid of the xeno demographic with not using heparin is appropriate.
Kim, from your perspective, what does the industry still need to do to make cell expansion more reproducible, also from lab to clinic? I'm thinking about, the fact that biological variability is still a major issue in cell culture.
I think really it's more access to raw goods in terms of being able to get the commercialization amongst different companies to a bigger spot.
And what changes when a company moves from research-grade materials to GMP-grade materials for clinical development?
So ours specifically, there's not too much of a change between the research grade and the GMP. So when different customers are moving into those clinical trials, there's less testing that needs to be done for our products. So it's pretty translational. They can move right along seamlessly into the different product set that they need.
And of course, we are here in Dublin at the ISCT. Kim, from the conversations you're having, do you think that people are thinking about cell conditions enough?
So I think what's happening, a lot of people are already in their clinical trials, and it's hard to change once you already have your product sets out there. So they're really stuck on what they're currently using instead of looking into the future as to what may better their cells in terms of growth and productivity over time. So I think starting to learn about the different medias that are available to them, why each of them is a little bit different. So again, ours is fibrinogen depleted and how that may help their cell growth versus somebody else's product line. So maybe customers thinking a little bit more along the lines of what is the actual difference between HPL versus serum versus other commercially made materials out there?
And just to sum it up, for a therapy developer choosing between culture material suppliers, what should they be looking for apart from the price?
Really, what's going to be the most efficient and effective product that's going to grow their cell types. Ours grows with like I said, the NK killer cells, the T cells, MSCs, fibroblasts. There's a wide range that a lot of the HPLs work with. but trying to narrow down what's going to work the best for them.
Well, Kim, thank you very much for joining us,
Thank
for
you.
your time.